Cy the Cyanobacterium, written and illustrated by Caitlin Evans. It was late summer and Rory was hot. She set off for her favorite swimming hole to help her cool off. It was a serene lake, typically blue and glassy, yet when she arrived, it appeared quite grassy. Green and fuzzy, the lake seemed to be covered, and when she knelt, a new organism she discovered. "Hi, I'm Cy," the blue-green prokaryote proclaimed. I'm a cyanobacterium, a relatively new name. I was once grouped with algae because I have chlorophyll, yet with further observation, I don't quite fit that bill. I have mesh-like peptoglycan in the walls of my cells. I have a lot going on, but no membrane-bound organelles. I'm a photoautotroph, converting light from the sun into carbohydrates for energy and oxygen for everyone. I also fix nitrogen. You see, I'm quite clever. The key is preventing oxygen and nitrogenase from coming together. I can work quickly, form heterocyst, or fix at night, turning an essential element into usable forms for other species to bite. It's been said I partner up with filamentous fungi and yeast. Together we form lichen, which clean the air that we breathe. Like any living things, I have good qualities and bad. Warm temperatures and stagnant water provide a lot of nutrients to be had. I eat, growing, dividing, multiplying. Eutrophic lakes like this one have a lot of nitrogen and phosphorus supplying. I can edge out other organisms with the propensity to consume. And before I even realize it, I've become a cyanobacterial bloom. You might hear bloom and think, oh my, how pretty. But I tend to choke out other life. It really gets quite gritty. The problem is, once I've bloomed, I hold toxin within my cells. And after blooming, my cells can burst and release it into wells. Contaminated water presents a global problem. So the least I can do is offer tips to help the future solve them. Farming is a pretty big deal. The demand's high, so we've turned to fertilizer, yet regulating it would be enormously wiser. Be mindful of your water use and gardening at home, and kindly, please clean up your pet waste. Don't just let them roam. If nothing else, knowledge is your greatest power. Thanks for getting to know me now. I've got some inorganic nutrients to devour. Rory, still quite hot from summer, had hardly stopped to blink. She couldn't believe this microscopic bacterium had really made her think. She turned around from her swimming hole, but rather than just mope, she decided to cool off with a popsicle, her new knowledge providing hope. She walked and thought about just how many Cys were on that lake. If something so small could have such an impact, what changes could a kid her size make? Author's note. Hi, I'm Caitlin. I have a 2-year-old daughter named Aurora, Rory for short. I spend hours each week reading books with her, and I'm always so inspired by the way these authors take complex concepts and make them digestible and entertaining for children. The addition of bubbles, balls, animals, or in the case of my book, popsicles, never hurt either. When I read over the guidelines for this term project, I couldn't resist the challenge of writing a rhyming story about cyanobacterial. As a young girl, my family would spend 2 weeks camping at a remote lake in southern Oregon. One summer, dubbed Algaean Summer by my best friend and I, the lake was unswimmable because of a toxic algae bloom. Over the years, the conditions of this lake worsened and native fish and other wildlife couldn't survive. Unfortunately, the lake had to be drained and restored over the course of years. For a long time, we didn't see the freshwater otters that used to live around the lake, but just last year, 25 years later, they returned. The illustrations in this book are my own and therefore a little rough around the edges. But what I hope everyone, kids and adults, take away from this story is that there's good and bad in everything. Different circumstances or situations can bring out our toxic sides, but learning more about these situations can make a big impact. Microorganisms teach us that our size doesn't determine our mark on this world. We can all make a difference. The end.
The bacteria that I will be covering today is Vibrio parahemolyticus. Vibrio parahemolyticus is the world's most infamous cause of foodborne illness. It is closely associated to shellfish, and it gives symptoms of acute gastroenteritis. When we think of illness related to oysters, it is likely from Vibrio parahemolyticus. In the following slides, I will cover morphology and phylogeny, as well as the main variables for survival, which include virulence factors, biofilm formation, and antibiotic resistance. I will also cover interactions with other microorganisms and the predator and prey relationships that they have. Vibrio parahemolyticus is a part of the Vibrionaceae family. Within the Vibrio genera, there are 40 species, and some notable relatives are Vibrio cholerae and Vibrio vulnificus. Vibrio parahemolyticus is gram-negative, bacillus, slightly curved, flagellated, facultative, anaerobe, halophile. There are two different types, swimmers and swarmers. Swimmers have a polar flagella that allows them to swim through water, and swarmers have lateral flagella, which allow them to move through more viscous substrates. They are considered to be halophiles because they prefer to be in saltier water, about 20 to 25 parts per million salt concentration, and also warmer water as well. Some of the virulence factors that Vibrio parahemolyticus possess are multivalent adhesion molecule 7, or MAM7, which allows them to form biofilms. It is also a key component in cell-to-cell attachment, which they utilize for infection. They also utilize type 3 secretion systems and various gene-related toxins like thermostable direct hemolysin and TDAH-related hemolysin, as well as antibiotic resistance that I will talk about in the next few slides. Type 3 secretion systems are essentially bacterial organelles that act as needles to directly inject bacterial proteins into a host cell. So in this image here, you can see a bacterial cell injecting itself with the T3SS into a different cell. These secretion systems are the primary mechanism of infection, and they affect the host cell's ability to produce energy. This ultimately leads to the loss of overall cell viability. TDH and TRH, or thermostable direct hemolysin and TDH-related hemolysin, are genes that enable Vibrio parahemolyticus to lyse the host's red blood cells. This essentially means just destroying those red blood cells. TDH is a pore-forming protein, so once the bacterial cell has infiltrated the host cell, TDH will come in and widen the pores on the host cell's mitochondria. This affects the host cell's ability to produce energy. TRH can change the host cell's morphology in a variety of ways via disintegration of the nucleus or the cytoplasm, as well as external structures like microvilli. Another virulence factor of Vibrio parahemolyticus is antibiotic resistance. There are actually numerous antibiotic-resistant strains of Vibrio parahemolyticus that exist in the environment, and this is due to the misuse of antibiotics in human and also aquatic animal infections. This is important to consider because Vibrio parahemolyticus can transfer these resistant genes to other human pathogens. Vibrio parahemolyticus have a variety of interactions with other microorganisms. For example, it has a commensal relationship with zooplankton, which basically means that Vibrio parahemolyticus benefits from the relationship, but zooplankton is neither harmed nor benefits. Vibrio parahemolyticus utilize the plankton for protection and also transportation through the water column, and they're often called the hitchhikers of the bacterial world. On the opposite hand, we have Halobacteriovorax, which is actually predatory to Vibrio parahemolyticus. Halobacteriovorax occupies the periplasmic space of the host cell and uses the cytoplasm for nutrients until it can lyse out of the cell and spread more infection. Researchers are actually trying to figure out how to utilize Halobacteriovorax to minimize populations of Vibrio parahemolyticus. And here's my reference list. Thank you for listening.
For this presentation, we're looking at the microorganism Helicobacter pylori. Helicobacter pylori is a gram-negative, non-fermenting, non-sporulating microaerophilic bacteria. It's about 2 to 4 micrometers long with a diameter of about 1/2 to 1 micrometers. It has a helical shape with about 2 to 7 Lophotrichus flagella at the end. That means that all the flagella are used for movement in one direction. It does come in a couple other shapes, but the other shapes are not as useful, just in rods or curved. The spiral shape is the one that's most successful and impactful. Helicobacter pylori is within the bacteria domain, and as you can see on the slide, it shows the phylum, class, order, family, and then the genus and species, which is Helicobacter pylori. This microorganism is a highly successful and prevalent bacteria. I saw a variety of different estimates in terms of the prevalence, but one that I saw is approximately 2/3 of all people in the world are believed to have been infected at some point, with higher prevalence in the developing world. The vast majority of people who have this bacteria are asymptomatic. but those who do have symptoms can develop chronic gastrointestinal symptoms. H. pylori is believed to be linked to approximately 89% of gastric cancers, 5.5% of all cancers worldwide, making it the only bacterium linked to development of any cancer. H. pylori has an extensive genetic diversity of strains, which helps it thrive. So within one person, there can be a lot of strains, and between different people. The genus Helicobacter consists of over 20 recognized species, with many awaiting formal recognition. In addition to a lot of diversity within humans, there are related Helicobacter species that have been identified in the stomachs of other wild and domesticated mammals. There are a number of features that allow this bacteria to survive and thrive in stomachs. It's microaerophilic, so within the stomach, it only needs a little bit of oxygen, and it contains hydrogenase, which it uses to oxidize hydrogen in the surrounding environment to get its nutritional energy. From that hydrogen, it produces oxidase, catalase, and urease. The urease production is key to its survival because it takes it in, breaks it down, and it creates ammonia and carbon dioxide. That ammonia in the surrounding area increases the pH, making it more basic, less acidic, and creates kind of a window of less acidity right around the organism, which allows it to live. When people have an H. pylori infection, it impacts the entire microbiome in a person's stomach. It can be around 40 to 90% of all the microbiota when it's present. So it reduces the overall diversity. The change in pH impacts the ability of other microorganisms to survive. And then another way it can impact the stomach is that eradication therapies can also further ruin the microbiome. It uses its helical shape and flagella to burrow into the mucus lining of the stomach. So it gets through the mucus and up to the skin layer. Once it's in the epithelial layer, it produces adhesins which allow it to stick there and not get washed away. Once it is up against the stomach wall, it stays there and produces ammonia, which decreases acidity. The ammonia allows it to stay there, but the ammonia also is toxic to the epithelial layer, so it ruins the stomach lining. It creates an immune response, more acids. So basically, it sits there against your stomach wall, and then your stomach acid ruins your stomach lining, creating ulcers. With an H. pylori colonization or infection, people can experience abdominal pain, bad taste in the mouth, nausea, heartburn, belching, and the infection is related to a number of diseases: chronic gastritis, peptic ulcers, gastric adenocarcinoma, gastrointestinal reflux disease, and cancer. H. pylori has been isolated in saliva, gastric mucus, dental plaque, and feces. It's transmissible through oral route or fecal oral route. Right now, there are no vaccines, so hand hygiene, access to clean water are really important. There are a number of different antibiotics that can kill an H. pylori infection or colonization. Other therapies like proton pump inhibitors, bismuth, and a number of different foods are recommended. And if you're having all of the symptoms in the previous slide, it might be worth going to a doctor and getting checked out.
Hola a todos. Para mi trabajo de final de trimestre he elegido hablaros sobre Helicobacter pylori. Es una bacteria en forma de espiral que infecta el estómago humano y que es la principal causa de gastritis y úlceras. A pesar de su pequeño tamaño, tiene un gran impacto en la salud humana y tiene la capacidad de sobrevivir en ambientes extremadamente ácidos como es el estómago humano, y es algo que la hace única. En cuanto a su anatomía, es una bacteria, como ya he dicho, en forma de espiral. Presenta flagelos, lo que le permite moverse a través del moco gástrico, y su membrana externa está compuesta de lipopolisacáridos y proteínas adhesinas que le ayudan a adherirse a la mucosa del estómago, como ya hablaré más adelante. En cuanto a su filogenia, pertenece al filo Proteobacteria, a la familia Helicobacteraceae. Es un pariente cercano de Helicobacter hepaticus y Helicobacter felis, que también pueden afectar a animales. He querido hacer una novela gráfica donde represento, pues, dónde se crea la bacteria, bueno, dónde crece, a qué parte del cuerpo afecta. Como vemos aquí es el estómago, y en esta parte de aquí pues explico su metabolismo y la manera en la que crece dentro de nuestro estómago. Como ya hemos dicho y como todos sabemos, el estómago es un una parte de nuestro cuerpo con un pH muy ácido en el que no muchas bacterias pueden sobrevivir, y esa es una de las cosas que hace única esta bacteria, Helicobacter pylori. Lo que hace es secretar ureasa, que es una enzima que descompone la urea en amoníaco y dióxido de carbono, y esto lo que hace es que le ayuda a crear alrededor de ella un halo que lo protege de esa acidez. Los flagelos, como ya hemos hablado también, le ayudan a moverse a través de como las capas del estómago, a través de la mucosa, y le ayuda como a buscarse una parte en el estómago donde ella se protege y pues puede crecer y desarrollarse ahí. También es importante saber que es una bacteria microaerófila, lo que significa que necesita muy bajas concentraciones de oxígeno para sobrevivir, y que su metabolismo se basa en aminoácidos, especialmente el glutamato y el aspartato, en lugar de los azúcares, que esa es otra de las cosas que le permite vivir en el estómago, ya que a diferencia de otras bacterias que utilizan más los azúcares, ella utiliza los aminoácidos, y el estómago es un lugar donde no hay tanto acceso a azúcares. ¿Qué más? Las interacciones con otros organismos, pues como hemos dicho, altera la microbiota gástrica al competir con otras bacterias por el espacio en la mucosa, y en las personas infectadas la diversidad de la bacteria pues se ve reducida, y eso favorece a un ambiente de inflamación que va a provocar esa reacción de nuestro sistema inmune. Cuando H. pylori coloniza el estómago, se activa esa respuesta inmune causando la inflamación crónica, y eso pues atrae a todas esas células de nuestro sistema inmune que están diseñadas para defendernos de esos ataques. Pero H. pylori ha desarrollado, ahí veis que tiene como un pequeño antifaz como para esconderse de esos linfocitos que nos protegen de infecciones, y es porque ha desarrollado esa habilidad de esconderse o de evadir el sistema inmune, y es modificando las proteínas de su membrana para no ser reconocida, básicamente. El impacto impacto de la salud de esta bacteria está asociada con gastritis crónica, úlceras péptidas y un mayor riesgo de cáncer gástrico. El tratamiento consiste en como 3 diferentes partes. 2 de ellas son antibióticos que van a, como serían la claritromicina y la amoxicilina, que lo que van a hacer es destruir la bacteria interfiriendo con la síntesis de proteínas y la pared celular, y por otra parte un inhibidor de la bomba de protones para reducir la acidez del estómago, debilitando la bacteria todavía más. No hay una manera de prevenir esta infección a través de esta bacteria, o sea, no hay una vacuna, no hay una manera de prevenirnos artificialmente o de buscar esa inmunidad artificial a través de una vacuna, pero sí que está la posibilidad pues, de prevenir esa infección a través de buenos hábitos higiénicos, lavarse las manos, cocinar la comida totalmente, beber agua potable, etc. Entonces, como conclusión, podemos decir que Helicobacter pylori es una bacteria que está increíblemente adaptada para sobrevivir en un ambiente hostil como es el estómago, y la capacidad para evadir el sistema inmune y causar enfermedades graves es un tema de gran interés en el mundo médico. Espero que esta presentación os haya ayudado a entender mejor su impacto en la salud, y gracias por verme.
You go to the hospital because you had stomach pains. You have a cut in the skin that you didn't notice, but there are loads of pathogens at the hospital that enters through the wound. A few days later your symptoms worsen and you return back to the hospital. Your doctor says you have MRSA. But wait. What is MRSA? Before we answer those questions, let's rewind. Hello. I am Staphylococcus aureus. You've probably heard of me. Like the others, I'm a gram-positive bacteria and a facultative anaerobe. I have a coccus shape that's arranged into clusters resembling a grape. I am part of the Firmicutes phylum and Bacilli class. Hello. I am Staphylococcus epidermidis, and I make up the major skin flora. I'm an opportunistic pathogen. I help the skin by preventing overgrowth by competing for resources, nutrients and space. I form biofilm to control desiccation and protect others. Anyways, back to me. I also live on the skin and mucosal surfaces. I am resistant to radiation and desiccation. I love living in high saline conditions of up to 28% sodium chloride. I have highly crossed peptidoglycan in my cell wall that protects me. As a facultative anaerobe, I can live in both aerobic and anaerobic conditions. I mainly prefer glucose as my energy source. I can use a variety of different electron acceptor including O2, nitrate, or nitrite. I can also use fermentation if I'm stuck and produce lactic acid. I live in a wide range of temperatures and even withstand up to 60 degrees Celsius. I like it hot. Now what would happen if I entered through the skin? I can evade your immune system by using my capsule and protein A to disrupt opsonization and inhibit phagocytosis. I have plasmids with antibiotic-resistant genes. I produce beta-lactamase that inactivates beta-lactam antibiotics such as penicillin. I have pathogenicity islands that contain genes coding for toxins, adhesins, and immune evasion proteins. I have transposable elements. These are genetic elements that can move within the bacterial genome. This comes in handy when I cause toxic shock syndrome. A little peek for what's to come. I produce coagulases that affect blood clotting. This allows me to be highly adaptive and a dangerous pathogen. You can get me from many exogenous sources including people, objects such as dishes or clothing, the environment, and occasionally animals. I cause 3 types of illness. Pyogenic infections. I cause localized abscess-like styes and boils, deep-seated infections such as osteomyelitis and endocarditis, and more serious skin infections like furunculosis. Toxigenic infections. I produce TSS toxin that grows in the wound or vagina. TSS toxin absorbs into the bloodstream leading to toxic shock syndrome. Foodborne intoxications. I grow on food that has been left out for too long and produce enterotoxins. When consumed, the enterotoxin enters the blood leading to food poisoning. The reason I am most dangerous is my ability to be resistant to antibiotics like penicillin, methicillin, and other common antimicrobial drugs. Now let's get back to you. We last asked what is MRSA. It stands for methicillin-resistant Staphylococcus aureus. Remember my plasmid. It has antibiotic resistance genes that codes for beta-lactamase. This inhibits beta-lactam antibiotics such as penicillin from working. Methicillin is a semisynthetic antimicrobial drug that was produced to fight back against me. But it lost once I developed antibiotic resistance genes against this. I am just too good where I have multi-drug resistant strains. The last resort is to use vancomycin to treat MRSA patients. But I'm starting to develop vancomycin resistance as well. Unfortunately, I lost today's battle. The doctor treated you with vancomycin and you started feeling better. You were discharged to go home and rest up.
Hi, this is my term project, and the microorganism that I chose to do was Enterococcus. An introduction about Enterococcus bacteria is that they are gram-positive lactic acid bacteria. They are facultative anaerobes, and they are most commonly found in the gastrointestinal tract. They can also be found everywhere else in the body, such as the oral cavity. On their shape and anatomy, they're usually oval, ovoid shaped, like in this picture here, and they're arranged in short chains of pairs or sometimes single cells. They're unencapsulated and they have thick peptidoglycan walls because they are gram positives, so they just have thicker walls than gram negatives. And they consist of teichoic acid and polysaccharides on the outer layers of the walls. A little background on Enterococcus is that their phylum is Bacillota, and then the class is Bacilli. They were originally named after Streptococcus, but they found that certain Streptococcus bacteria were a little bit different, so they decided to name these bacteria Enterococcus. And they're usually identifiable through genome sequencing and 16S rDNA sequencing. Their metabolism and growth, they use various metabolism pathways such as sugar, carbohydrates, citrate, glycerol, and fermentation for dairy products such as milk, cheese. And they like to grow in temperatures from 10 to 45 Celsius, and they can tolerate high basic pH levels and 6.5 sodium chloride levels. So they are very high tolerant to the environment. Now, for other microorganisms that I may interact with, some are including lactobacillus, which they can be found together in the intestinal microbiota, and they work together to improve digestion and reduce inflammation. Another one is Candida albicans. They are yeast, and they are usually found in the oral, intestinal, and the skin, and they work together to form a biofilm of oral cavity and prevent infections. For the environment and organisms, they are ubiquitous bacteria, so they can be found mostly everywhere. Found in places like humans, water, sand, soil, plants, vegetation, animals, and more. They're also most common in hospital settings, and they're hard to get rid of because of how resistant they become over the years to antibiotics. And this can also be due to healthcare workers not having proper hand wash techniques or other things, and this can lead to more infections. And then we have foods. So they are mostly found in a lot of foods, but for the majority, they can be found in fermentation foods, such as cheeses and fermented meats like fermented sausages. They're hard to kill off as they can resist high temperatures. Even if they go through a high technology heat killing, there's still some free floating bacteria. And they're low grade pathogens, making them cause food intoxication, which can be really bad on the human body. Lastly is my art project. I'm not really sure if you can see this well, but I did my art project in pencil and I drew the gastrointestinal tract here with a little bit, some microbes floating around. I also drew water and then some grass, which represents the water soil. And then there's a picnic plate here. Sorry, a picnic mat here. It has a piece of cheese on it, but I'm not sure if you can really see it. And then a tree. And then I drew it all pointing towards the Enterococcus microorganism here. So I drew a thick peptidoglycan wall, and then here's some teichoic acid on the outside, and then just a simple structure of bacteria. And that's basically everything for my term project. I hope you guys enjoyed it.